Okay, good morning everybody. So today I'm going to talk about different issues. We have a very wild season, 2016-2017, and this has provided us conditions to study aphid populations, insecticide use, and also biocontrol. So that was very interesting. And here you have a table of the insecticides that are used for foliar treatment of aphids and grasshoppers, armyworms, and also multi-active ingredients when you have more than one pest in your field. So a couple of things are interesting here. It's hard to read, but for aphids, we only have pyrethroids. That's the only product that we have. There are different compounds, but they are all pyrethroids. So resistance can be developed. They are not used judiciously. The other thing is for grasshopper, we have dimethoate. That's the only difference between controlling aphids and grasshoppers. So besides that, for armyworms, we have two products that are interesting. BT, that's a biological-based insecticide, and also we have here spinosad. Spinosad was based on a fungus, so that's very important. So remember then, for aphids, only we have pyrethroids. So what we have done since last year is we have a project with Dr. Carl Bradley where we are trying to see the population of aphids and the viruses that we can have in the fields. So far we don't have anything to report yet, but besides that we have a study in Princeton where I have two blocks of wheat. In each block we have a treated seed Pioneer, untreated seed, the same variety, and also we have two untreated varieties. So in those two fields, we have only spraying one of them to see if there was some aphid activity. And we did a spray just as a standard spray that you do here. So far, you can see here this graph. This is March 6 till 20 April, and this is the number of aphids per feet of wheat. So we don't have numbers that are above the threshold. The threshold is 10 aphids per feet. So we didn't have. So this is combining all these three plots. So we separate that stuff. We separate the varieties. The results are very similar. So we don't have that many. 3.5 is the maximum here. So two aphids per field, and so on. So they're not big numbers at all. So one thing that is important here is that sprays for aphids are based on economic, on the monitoring, and also on the thresholds. Oh, sorry. If you don't believe me, there is some people from other university. So this is Dr. Scott Stewart from the University of Tennessee, just south of us, and he said that both an insecticide seed treatment and late winter foliar insecticide application is not necessary if you plant by middle October or later. Even that means people in the south, Mississippi, Louisiana, and so on, they believe that you need to use a seed treatment, and one of the explanations for how these seed treatments work over there is because they have a sandy soils. It seems that sandy soils cannot interfere with the activity of the insecticide. In our case, we have a clay soils, and then that interferes with the activity of the insecticide when they are used for seed treatment. Besides that, I understand that growers here do standard applications when you try to control other insects such as armyworms. And for example, this year, I believe that armyworms were a fact because they appear very early. It was not reported before armyworms besides before March, but we have some numbers this year. And then it went up, and you can see by 27th of March, we have a very high numbers compared to the rest of the years, and even on 3 April, so the black one. But then it seems a decrease, and we have here on the kind of yellow, that's in 2008, we have a higher number. So in those cases, people do the applications. Also when people do the application for herbicides or for fungicides. So now, I believe that in spite of that, we have a crazy weather, and these have consequences for aphids, and not only for us, but for aphids. And I present this graph before, and this is the activity of aphids the aphids activity during the winter around 45 degrees or plus that. So based on that, we have for aphids that the winter during this season was 90 days compared to, this is a 20-year intervals, 1976, 1996, and you see in that first one, we have a winter for aphids that's 120 days. 20 years later, we have 110 days, and then right now we have 90 days. So it seems that this, again, this is a crazy weather, and this is data that I collected from the University of Kentucky. Besides that, also we have here the relative humidity. And I took data from 2010, 15, 12, and 17. And you can see there are two big differences here. The 2012 and 2017 have higher relative humidity than the other 2 years. So this is from January up till May. So this is important for what I'm going to talk to you later. Besides that, I thought that we have a very heavy rainfall. That was my impression. However, if you see this information, there is years before that we have higher rain activity than this year. So I thought that what I'm going to explain you later, it's going to be based on rainfall, but this one on the top, the relative humidity, was very important. And let me tell you, this is a very exciting information for me from the point of view as an entomologist, because I was working with a farmer here, and this farmer have larger numbers of aphids. One of the commentaries I heard is that they told me that they haven't seen this number of aphids in 20 years of experience. So this is the field, and in this field he flagged a square, and in this square he sprayed these insecticides. He sprayed on March 12, Belief, Indigo, Hero, Dimethoate, Declare, and so on, and Stallion. And when I visited him, because he was worried that this field maybe have resistance, has aphids that have developed resistance to the pyrethroids, because he uses pyrethroids twice on the fall and twice on the spring. So he used twice, and they weren't controlling at all. So he was worried that these aphids have developed resistance. So I purchased a product called Exponent. This is a piperonil butoxide, and this breaks the enzymatic activity of aphids that have developed resistance to pyrethroids. So I bought that stuff, and that's where we did a spray 2 days later. So what you can see here, that on April 13, there was a decrease of population of aphids in all treatments, even the ones that were not sprayed, because the control and the warrior here in black and the warrior with Exponent on this one was not a spray yet during that time, but there was a decreasing activity. So by the April 17, there were very few aphids, so only the control have higher numbers, and you can see here, this is representing what you have here. So there was a reduction on number of aphids only at the 5th day, so there was very few aphids. However, in none of these cases we have significant differences. Okay, so something else was happening in this field, and this is what I'm going to show you what. So we took some plants to the laboratory, and in the laboratory we picked up some aphids that we believe they were healthy, and then we put in the petri dish. We put 10 aphids, and we replicate 3 times, and then we were trying to do a LCD. There was a control. The control was no insecticide, only water. And then there was a Baytroid. We use Baytroid. These represent 2 ounces per acre. Okay, and these are the aliquots from this. So from this insecticide use. And we see here, this is the percentage survival of aphids at 24 hours. So you can see only the 19.77 rate had a 80% survival, whereas the lower was on the highest rate. However, still there was no significant differences on that. And right now, I cannot say if there was resistance of this population to pyrethroids. I cannot say that. So why is that there was a high mortality, 40% in the control? Something else was happening here. So besides that, we also evaluate the number of nymphs that these aphids were ovipositing. And that was interesting because there were fewer numbers on the higher rates. And probably there were, I believe that there were not resistance here. I'm not sure yet, but we still need to do that more work. However, 48 hours, there was 100% mortality across all treatments. And all the aphids, most of the aphids were in this stage. They were being killed by this fungus that was in this field. So you can see here, these are leaves. And I have a leaf here. I have a container here where you can pass it. So you can see the leaves there. And there's the fungus that was growing, but that was on the body of the aphids. Okay. So what else was happening here? Let's see. So we took samples like you can see there to the laboratory, and then we leave in those shoe boxes, and then we leave that for 4 days, and we see how many parasitoids were present there. And this is what we found. We found that the warrior and the warrior plus exponent, it has a high number of parasitoids. There were bundles of 40, 50 stems per these shoe boxes. And you can see there were significant differences also. So there were more on the warrior plus exponent, and in the control, compared to the other insecticides that were multi-active insecticides. So two things are important here, two messages that you need to use. One is the use of multi-active insecticides judiciously. So you need to be sure that you have the insect that is necessary to use this insecticide. And also we need to be aware of the natural enemies. So I cannot predict, and we cannot predict, what's going to happen next year. Why is that we have this entomopathogenic fungus this year? It's probably to that relative humidity, but we are not so sure. So when I was in this field, I was lucky. It was lucky for me that I talked with the farmer, and I told them that we have this kind of aphids. And you can see there were some white spots in the aphids. So this indicates that there was a presence of fungus there on the aphids. And this is what did happen probably 1 week, 2 weeks later. All the populations on these two fields, there were two fields. One was in Logan County and the other was in Hopkinsville. After 1 week, also most of the population of aphids were killed, so there was nothing. And that shoebox that I'm showing you over there, we collected that yesterday, and there were no aphids at all there. So you cannot find any aphids. So just only, you can see here, I have some mummies, and these are mummies that are produced when the small wasp comes and lays an egg on the second or third instar aphid. It doesn't lay eggs in the big females, it lays in the small ones, and it starts to develop inside the body. Then it comes out of that mummy later on. You can see here, there's a picture of this mummy. I'm showing you here, there's some mummies here and some very tiny aphids. I'm not so sure if you're going to be able to see, but while Carl talks, you can take a look of that. Any questions?